Emily, if you want to go, or
yeah, I'm fine. Could you repeat that question, please?
So you were asking about you presented about water bodies, physical chemical characteristics, right? So I'm wondering if you're planning to look at microbiota associated to that water bodies that enhances the physical chemical characteristics that you're analyzing. And in the case of Alicia, is there also can you give us a little bit more insight about your work about particular species or genus of a species that are related to these being more attractive to be bitten by mosquitoes?
Okay, thank you for the question. I'd look into the literature, and in fact, I think there's some literature that does relate some physical chemical parameters to micro compounds underwater, and that's highly related to pollution. But that goes beyond our knowledge in the research group. But it was indeed a question that we wanted to look for, but we did not have the required skills to conduct that study. But it was a good one, in fact. But thank you for the question. Isn't it linked with physical chemical parameters?
Great.
Thank you, Emily.
For my question, so from this study, we have candidate attractive and repellents that we found that are differentially abundant, but we can only get general level data because of the type of sequencing that we are using. And we had odor data available, so I used some predictive tools to try to predict candidate attractants and repellents, and then looked for the quantities of those in our data. But the next step would be to culture species within these genera and actually look at what attractants and repellents these bacteria are giving off, and then test that with the mosquitoes.
So I have a quick question for Alicia. So you haven't tested if these bacteria in culture are attractive to mosquitoes? Have you tested that?
No, we have not tested that, but there are some studies from Verhoef et al. That did do that.
Okay, good. Is any other question to any other of our participants? I don't see anything on the chat. So, Andrea, I think I'll ask just the devil's advocate. Just so, do you think it could be anything wrong there on the technology that you're using that could just jump into a species or cause these effects on other untargeted species, even though like if the eggs are being released and you have no female mosquitoes, I think that's always the concern with you're muted.
Yes. So basically the gene femalis itself is only found in anophelines. It's like a very, very distant homologue to transformer 2, but like very distant. So it's really, I mean, the intention of the technology is to release basically GM male mosquitoes, right? So anywhere that these males can hybridize, right, and form fertile offspring, the transgenes could get into, right? So the males that we're releasing are transgenic with two different transgenes. If you decide to let them stay fertile and have just female suppression effects, then they are technically transmitting transgenes through the population, albeit non-driving ones. However, the more kind of clear immediate use is in SIT, right? Using some of the sterilization protocols that we have for chemo and radiosterilization. So they're standard transgenes, right? They're not even gene drives. They're going to be subject to normal Mendelian inheritance. In fact, I mean, right, the canon in the field is that all transgenes in anophelines make them slightly less fit. So we do actually expect these transgenes to kind of taper out over time. Yeah, it's just a very powerful system. And it really does seem like the only times we've seen anything that looks even remotely like a female is that they're genetically either XY, or we did to, in the interest of time, I left this out of the talk, but after multiple generations, at about generation 4, you do start to see what looks like a very rare resistant allele pop up that allows at least some fertility, or sorry, some viability of females. But these females are dying during larvahood. So they don't even really make it to pupahood, let alone fly away. And in the times when we do see like an XY female, they generally have flight issues because their flight muscles aren't kind of perfectly sexually tuned for their body weight and body dimensions, because their flight muscle morphology is also subject to kind of sex-specific signaling. So, I mean, never say never, right? It is biology, but it's a very surprisingly strong, powerful phenotype. And the nice thing is you maintain these lines completely separately in the lab until you're ready to cross them together. So unlike gene drives, there's really no chance for resistant alleles, or at least they don't make their own resistant alleles as you maintain them in the lab, because until you're ready to release them, the Cas9 and the gRNA are kept separate. So never say never, but it's
are there
cool
any progress on or already steps working on the implementation of this on the field?
Not yet, no. So we've really just submitted. The paper's under review with scientific advancements, I believe, or scientific reports. And it's on bioRxiv if you're interested in the manuscript. But no, we haven't progressed to the field. The Akbari lab doesn't really have too much expertise in true translation into the field beyond what kind of biotech connections we have. And so I encourage anyone who's interested in maybe helping us initiate that, or anyone with field experience, please contact Omar Akbari at UCSD, and feel free to start that conversation, because the technology is there and ready to go.
Well, we seems to be some important people for those things in this audience, like Henry.
Yeah, exactly.
There. Any questions? Any other questions from the audience?
Yes, please. I have a question for Aisha.
Yes. Okay, please.
Okay. Hi, Aisha. Nice presentation. I want to know how many days were the mosquitoes that were fed on the infectious blood maintained for? And then is it the same mosquitoes that you dissected that you also did the PCR on, or a different cohort? If you had different cohorts, or I mean, you feed the same batch and you dissected some, and then use the different the remaining ones for the PCR and the sporozoid confirmation. Thank you. I don't know if you get my question.
Yes, I'll answer your question. So for the mid-gut
Okay.
dissections, we maintain the mosquitoes for 8 days post-infection feed. And then once we got confirmation that these mosquitoes actually had oocysts that were prevalent in them, we then took the rest of the samples and maintained them for 16 days. And no, we actually divided the samples. So those that did mid-gut saliva gland dissections, we had a sample size for qPCR and a sample size for ELISAs. And the results we had from that, we then compared. We didn't actually use the saliva gland we dissected for qPCR. We just had different samples for each assay. But it was from the same batch of infectious mosquitoes that we had. I don't know if that answers your question.
Yeah, yes, please. Thank you. Before, I thought you compared, as in the same sample that you used for the oocyst dissection, that's the mid-gut. Then you later, that's keeping them more than, say, 10 days. Then you dissected the gut and also use the head region for the ELISA test. But this explains it. Thank you.
No problem.
Any other question? No? Okay. Well, thank you very much, all presenters and audience, for this wonderful session. We welcome you like to stay tuned and welcome back again for the latest part of the malaria symposium. Thank you very much, everyone.